So thank you for the introduction, and thank you for the opportunity to present. And good morning, everyone. So the outline of my presentation is I'll first talk about the impact of carbohydrate availability and allocation on fruit set, talk about flour quality and how that impacts fruit set, and then talk about the limitations associated with fruit set, and then finish off with some knowledge gaps and some potential intervention. So I'll first start off with some background about avocado reproduction. So fluoroinductive cues produced in leaves, when they travel to the buds, they can induce inflorescence development, and during that period you get flower development. Flower quality, which I'll talk about in a few slides, is determined some point during flower development, and when that ceases, you can then have fruit set. And there's a number of factors required for fruit set, from pollination to fertilization. Okay, so fruit set really by definition is something when the fruits begin developing, and it's during fruit development that you can have abscission occurring. Also, the other thing to consider with regards to the reproductive cycle is the previous year's crop, not only how much crop the trees are carrying, but when that crop is picked. And then the environment. So a lot of these processes are impacted from environment, and I'll talk about that. All right, so carbohydrates, or non-structural carbohydrates to be more specific, are produced by leaves in the canopy via photosynthesis. When photosynthesis produces carbohydrates in excess, they can be stored in woody tissues as reserves. And so carbohydrates in general can be leveraged either from photosynthesis or from reserves in avocado. And that can depend greatly upon the time of the season. Work in the literature suggests that carbohydrates are critical for flowering, fruit set, and fruit development. So work that we did in AV23010 indicates that stem starch levels at bud burst is a good indicator of the level of flowering. It's a good predictive indicator. Work from Inaki Hermaza in Spain. His work indicates that the flower carbohydrate status is a good indicator of fruit set. And then work that we did in AV26005 indicates that the carbohydrate status of the seed coat, that is the maternal tissue of the seed, determines whether or not a fruit will develop to maturity or whether or not it will upsize. So fruit set occurs during flowering, and quite often flowering can be quite profuse in avocado. And so the prevailing hypothesis is that the high intensity, when avocado flowers at high levels, that can drain carbohydrate reserves used to support fruit set and development. And so what this indicates is that avocado trees don't really have an apparent built-in mechanism to conserve reserves. And this likely reflects the fact that these trees are semi-domesticated at best. And so they're really genetically programmed for survival and not production. And so if one wanted to manage carbohydrates in this sense, the way to manage it would be to somehow, one could regulate that flowering intensity and reduce it, that may free up carbohydrate levels to support adequate fruit set and fruit development. So avocado undergoes multiple cycles of vegetative growth during the growing season, so it has a bias for vegetative growth, and that can be a considerable challenge for canopy management. And also, the overlap between vegetative growth with reproductive growth is thought to be a major contributing factor for the low production potential of avocado. So for example, there's some experimental evidence suggesting that the outgrowth of the spring flush during flowering reduces fruit set and can lead to early fruit abscission. And there's some hypothesis that the outgrowth of the summer flush can impact the summer fruit drop. So here, allocation or partitioning of carbohydrates to support vegetative growth then limits reproductive growth, both fruit set and abscission. So we can ask, can annual yields be increased by reducing the expansion of the spring and summer flushes, or particularly the spring flush? And so, oh, that's pretty bad. Anyways, you don't really need to read the details. I'll just summarize it. So there's a number of publications that came out from the late 80s to the early 90s, looking at the impacts of paclobutazol, an inhibitor of gibberellin biosynthesis that's used to reduce shoot growth. And what was found at various concentrations applied to either fuerte, Hassan fuerte, or Hass by foliar spray, applied sometime around flowering, that when this applied, in all cases, this reduced the length of the shoots. In a few of these publications, they also reported this reduced leaf area. This publication here in blue reported that there was an increase in fruit set, a reduction in early fruit abscission. However, looking at the yields at the end of the season, there was no difference because the trees had adjusted the crop and dropped the fruit in the summer. There was one report from Avocado in Israel that reported that applying paclobutazol reduced for early fruit abscission, and that led to an increase in yield. Tony Wiley also reported that applying paclobutazol also increased yield, although he didn't look at these yield determinants such as fruit set and fruit abscission, and that this increase in yield wasn't due in part to increase in fruit size. So the summary of this work indicates that paclobutazol suppresses the expansion of the spring flush, but it also reduces the leaf area. And the results from here are quite variable in terms of yield. And that could be due to the location, the environment in which it was applied at, concentration, and timing. And so I think maybe to get a more robust impact, taking a PGR-based approach, maybe what needs to be done is to apply not only growth regulators to reduce the growth potential of the vegetative shoot, but also increase the potential for fruit set in the flowers. So other evidence to support that allocation of carbohydrates impacts fruit set as well as abscission comes from looking at indeterminate and indeterminate inflorescences. So avocado can produce both of these. So determinate inflorescences are ones that end in a flower, and indeterminate inflorescences are one that ends in a vegetative bud. And it's this bud that will grow out and form that spring flush. And what's been found is that determinate inflorescences are more productive in that they have a higher rate of fruit set and a lower rate of fruit abscission than indeterminate. And in fact, work that we did in AV23010 indicates that if you remove that bud and try to mechanically convert it to a determinate inflorescence, after removing that bud, those inflorescences will set and retain more fruit. However, with all these sorts of things, there's going to be a limit. So this is some photos. Oh man, the screen's terrible. Anyways, this is a photo I took this season out in WA. These trees flowered quite profusely. They had very little crop the year before. And most of the inflorescences they produced were determinate inflorescences. And so while they initially set a lot of crop and retained that, what happened is that with little canopy from the spring flush, most of these crops dropped, or most of these fruits dropped. And so in many cases, many of these trees dropped about 90 to 95% of their crop. So once again, this brings about the point that with avocado, it has a bias for vegetative growth, but the idea here is not to eliminate that growth, but to create a balance between reproductive and vegetative growth. So now I'll switch gears and talk about flower quality, which is a term coined by Inaki Hamaza in Spain. So before I get in that, I'm just going to go over some basic terminology regarding flowers. So the female reproductive structure is in the center is termed the pistil, and it consists of the stigma, style, and ovary. And inside that ovary in avocado, there's a single ovule with a single embryo sac. The male reproductive organs or the stamens contain the filament and anther. And it's the anther that produces pollen. And here, after fertilization, the embryo sac or embryo will give rise to the seed, and the ovary wall here will give rise to the periocarp. So one of the questions that Anaki was interested is trying to come up with a parameter to better understand fruit set and flower quality. And so what he found was that starch was highly variable in the pistil cells. And so some flowers, the pistils had relatively high starch levels, and some had low starch levels, and some had starch levels in between. And what he found was that in general, pistils that tended to have higher starch levels had a higher rate of fruit set and fruit retention than those that had low starch levels, which had low fruit set and low fruit retention. So we can ask, why are starch levels important for fruit set? Well, really, they provide an energy source. So an energy source to maintain pistil receptivity and ovule viability, pollen tube growth, and it's also probably important for kind of setting off fruit set and early stages of fruit development. So some of the knowledge gaps that still need to be completed here or looked at is we still don't know when and what controls starch accumulation in the pistils. We don't know what impacts temperature has on this, particularly cold temperatures, and whether or not there's other additional factors that determine flower qualities, such as hormones. Okay, so now I'll switch gears and talk more directly at fruit set. So going through some background. So fruit set begins with pollination. So pollen grains must be deposited on the stigma. You then get pollen germination, followed by pollen tube growth down the style, around the ovary, and then you get penetration of that pollen tube into the ovule, and you get double fertilization, whereas one of the sperm will fuse with the central cell to form the endosperm, and the other one will fuse with the egg cell to form the embryo. So fruit set can actually be initiated as early as germination. So in this case, that can stimulate cell division in the ovary wall, but you really don't get full fruit set until you get fertilization. And the reason for this is that the seed is really the hormone factory in avocado, and those hormones are essential for promoting fruit growth and development. So now I'll switch gears and talk about pistil receptivity and why this is critical for fruit set. So avocado flowers undergo 2 phases of development. So first they open up as a female, and during this stage, the pistil is receptive, but the stamens and pollen have not matured. After a few hours, those flowers will close. In a later time point, the flowers will reopen as a male, but in this case, the pistil is no longer receptive, but the stamens and pollen have matured. And so what happens from this transition from female to male phase is that pistil receptivity declines. That reduces self-pollination and promotes outcrossing. So outcrossing is really something that you see in wild tree species. So if one could manipulate avocado and have self-pollination, that would be a great benefit. And so as a result of this flowering phenotype, avocado requires insects such as bees and flies for pollination. So what we know about the physiology when flowers transition from male to female is that you get, in the pistil, you get starch degradation, you get the deposition of callose, which reduces the pollen tube growth, and you get ovule degeneration. There is some evidence in the literature that boron can increase fruit set and pistil viability and ovule viability, but it's likely going to have an effect in areas where boron is limited in the soil. From talking to Inaki Hormaza in Spain, he's indicated that increasing humidity can be a way to increase pistil receptivity. So I think one of our research gaps here is having some sort of knowledge base that we can leverage where growers could apply something to increase or maintain pistil receptivity as flowers transition from male to female to try to promote more self-pollination. All right, so now I'll talk about the impact of temperature and humidity on fruit set. So once again, the early events are diagrammed here in this image where pollens get deposited, they bind to the stigma, they then hydrate with water, they undergo germination, penetration, and pollen tube growth. And we know from the literature is that humidity tends to affect these early processes, and temperature tends to affect these processes here related to growth. So work from Inaki suggests that the optimal conditions for fruit set is temperature range between 20 to 25 degrees Celsius and humidity around 75%. So one of our gaps in this case is have a better understanding of how temperature times humidity impacts fruit set. If growers had a handle on this, or this could be better determined, then growers could look at weather forecasts and based upon those weather forecasts, maybe try to implement some sort of management strategies to maintain fruit set when you have poor conditions. So we can ask the question whether or not is pollination a limiting factor for fruit set and avocado? And so we know from work out of Israel in the late 80s that their fruit set required at least 20 or more pollen grains to be deposited on the stigma. Now, a number of studies have been done in California and in Spain, and I'll show you the one in Spain here. This is looking at the percent of flowers with 0 pollen grains, 1 to 5, and 6 to 10, and so on. What's found is at the female stage, shown here in red, that almost 80% of the pollen, 80% of the flowers don't receive a single pollen grain. So what that says is that most flowers don't receive pollen. And so Inaki looked at this further and asked, well, what happens if I now hand pollinate and now provide those pollen grains? What happens? And so what he did here was in the orange, he hand pollinated Hass with fuerte pollen, and he compared that to open pollination in the green. And he looked at fruit set over time in weeks. And so after 18 weeks, when he looked at the results, the open pollinated had about 0.15% fruit set, and the hand pollinated had 2.8, which is a 19% increase. However, you can still see here that you had 97% reduction in fruit set. Now, it's likely this is going to be attributed to flower abscission, which would occur probably in that first week, and then fruit abscission. So, this experiment illustrates that while pollination can be limited, there are other factors operating in the background, such as probably pistil quality, pistil receptivity, and fruit abscission. So concluding here, when looking at knowledge gaps, have a better understanding of how carbohydrate availability and allocation impacts fruit set and how that impacts flower quality and pistil receptivity. All that put together could be used to help develop, that knowledge base could be help used to leverage new tools to increase fruit set. Pollination, another key factor. So this is going to require pollinators. So having the ability, let's say, to modify nectar and make it more attractive could be a way to increase pollination. And pollinizers are also important in production. And then flowering is going to be an important determinant, as if you don't have any flowering, you're not going to get any fruit set. And then environment is also going to impact all of these processes. When it comes to management, right now, probably the best tool that growers would have to manage fruit set is going to be using PGRs such as paclobutrazol and or uniconazole. And once again, the idea here is to reduce the growth potential of the vegetative shoot, free up carbon, and then support fruit set and early fruit development. So some of the considerations here is concentration and timing. So for example, the question becomes does a grower want to apply a single dose at some early stage of vegetative shoot development, or does a grower want to apply lower doses but apply it maybe 2 or 3 times during that period of vegetative growth? Weather conditions is also going to be important. So paclobutazole and uniconazole not only inhibit gibberellin biosynthesis, but they also inhibit an enzyme that breaks down abscisic acid, which is a stress hormone. So if this is applied during a stress event, that could lead to elevated levels of abscisic acid, and by the name of it, abscisic acid can induce both flower and fruit abscission. And then also, it may vary from seasons to season. So for example, if a grower has a good level of flowering, high insect activity in the orchard, good temperatures, and if there's some indications you've got good fruit set, it may be a good idea to hold off on applying a PGR because these PGRs can inhibit leaf expansion, so reducing leaf area. So by holding off on that, you could increase the leaf area in the canopy to support that extra crop. One of the knowledge gaps that's really missing here is having a better understanding of the hormonal control of fruit set and its relation to flower quality and pistil receptivity. And the idea here is that if you have that knowledge, you can leverage it to try to develop a PGR-based application to target fruit set. And by combining a PGR that targets fruit set together with PGRs that reduce vegetative fruit growth, vegetative shoot growth, may be a more robust method to increase fruit set. And as I've already talked about, having a better understanding of temperature times humidity would be a very valuable tool. So for example, if the weather is forecasting for low humidity, if a grower only irrigates one time a day, well, maybe it's possible to irrigate multiple times a day to try to increase the humidity in the orchard. And then lastly, there's a number of new breeding technologies that are available. And the idea here would be to target and genetically improve Hass. So we know that there's been a number of breeding programs in the world, California and Israel and so on, that have produced new varieties, but none of these varieties have been able to displace Hass. So if you can't displace Hass, maybe the best thing to do is to see if these technologies can be used to try to make new clones of Hass that is better adapted to some of the environments that Hass isn't adapted to. All right, so just like to thank the people in the CSIRO Avocado research team and others that contributed to our work in abscission. The Avocado extension team for giving me the opportunity to present today. Growers and farm managers that have allowed us to go in and abuse trees in their orchards. Avocados Australia for their support, and Hort Innovation at CSIRO for funding. Thank you. Hi.
Okay, thank you. Thank you so much. All of those really interesting. Yeah, a lot of information was really great.
If you have questions, you can talk to me after.
Oh, yes. Yeah, we did talk
Oh, yeah, I do? Okay, all right. Any questions?
Questions?
Harley Thomas, have you showed a photograph of those trees in Western Australia that lost 90% of their crop? That's the question.
Can you repeat the question, please?
You showed a slide of the trees that dropped 90% of their crop in Western Australia. Did you take into consideration that in summer in Western Australia this year we've had 3 weeks of 40-odd degree heat with a relative humidity of about 10%? Because I followed that fairly closely, and I was budgeting on phone calls 10 days after that event. Guess what? On the Monday morning, when I got fruit falling on the ground.
Yeah.
As a result of that. So it was an environmental effect there that was great.
Yeah, but also when we monitored drop in those trees, we looked at the weather patterns as well, and you could see that some of that drop was occurring independent, likely going to be due to the fact that you just had very little canopy to support that crop.
We do give growers over there to irrigate things that operate.
Yeah. Yeah, this is at a well-managed orchard, so they really do. I think they over-manage their trees, but yeah, which isn't a bad thing.